I think we were just getting worried that we confused everybody.
You people are confused, but enough people are here.
We are. This is plenty.
The worst part was everyone I stopped to be like, how do I get over into the connector? They're like, I don't know.
Yeah, you never come to GSI seminar, right?
And certainly not at 11. So we're getting a whole new crowd. We're getting all the non-noon people.
Yeah, we did a 12 kind of nice time.
Although we are having lunch, if you guys can stick around, chat. Yes.
Hello? Oh. Yeah, you can just, I'll come downstairs. Once you come here, just call me. Okay. Oh, thank you.
I'm still sticking out my
Yeah. Are we waiting on Nelson?
Yeah, I think Nelson should be.
Everyone, while we're waiting for Nelson to get started, there's a QR code up on the screen. If you're interested in getting a copy of the slides from today's presentation, go ahead and scan this. I'll also show it again at the end of the presentation. It'll prompt you to fill out some information. It just lets me know that you would like a copy of the presentation. So thank you in advance for filling that out.
Say hello.
How's it going?
I can stay till 12, and then I gotta run to another meeting.
It's great to see you, Nelson.
Good to see you again too. Our admin Sabina is running the live stream. She said there'll be food coming around 11:30.
Perfect.
If you guys want to set up a table there and do lunch with anybody who wants to stay. Career advice or something.
Yeah. Awesome.
Yeah.
All right.
I'm gonna stay in the back, let everyone come.
Yeah. Thank you. Thank you, Nelson.
Yuri, do you want to do an intro and talk about the Instagram program?
Hello everybody, so my pleasure to introduce Juliana Crivella, field application scientist, who will be talking today mostly, as far as I understand, about the advantages of this relatively new kit that's called Flex. But before that, first of all, thanks to GSI for organizing this workshop and support BU cores as usual. Also thanks to the Next Genomics, who also provide some free reagents, which makes it possible. And going back to this Flex kit, I don't understand why people not yet using it more often, because it provides quite a few substantial advantages. Mostly it reduces the logistic difficulties, because you can now fix your cells instead of rushing them immediately to the core to run the 10x instrument. You can also multiplex several samples per lane, which reduces the cost. And also you require less sequencing in the end because it's probe-based. So we're hoping to provide some free pilot small projects completely funded through GSI and the Next Genomics. Illumina also might chip in, so the details are still vague, but something like small pilot projects with 4 samples should be plausible for you to do. So with that, Juliana, please.
Thank you, Yuri. Hi everyone, it's great to see you all here today. It's nice to see some familiar faces in the crowd. So like Yuri mentioned, I am your local field application scientist. So I'm here to help support the core and also to help support the end users with things like sample prep and troubleshooting. And like Yuri mentioned, we're doing a core lab grant program through the GSI where you can submit an abstract and you have the opportunity to win free reagents to do a 4-sample multiplex using our Flex kit. So the purposes of today's presentation are just to talk about what Flex, our fixed sample assay, is, and what are the different types of samples that you guys now have the opportunity to use when we're no longer boxed into using live cells for our assays. Okay, so for those people in the crowd who are new to 10x, we've been offering single cell resolution assays for about a decade now. And it's had a really amazing impact across all different fields of research, and it's providing the resolution to answer biological questions that we weren't really able to answer before. So while it's been great for us to provide those single cell assays for customers, there are obvious challenges to working with live cells. And really the first one, and Yuri kind of touched on this with his introduction, is flexibility. I'm sure there are many people in the crowd who have done a time point study. In fact, I've worked with some of you on your time point studies, right? And then you're sort of boxed into when your cells are ready from an experiment. You have to harvest them and then work with them immediately to get them into one of our live cell assays. This can really be a burden, especially if you're the grad student who's there harvesting cells at 11 o'clock at night, right? It's great to have the flexibility to choose when you run your samples through a single cell assay. The other thing kind of folded in with that flexibility is sample degradation, right? Most of us aren't using the common, easy to work with samples, right? Those are the low-hanging fruit that have already been researched pretty exhaustively. We're working on those niche tissue types, so those niche organisms where they really aren't happy in a single cell suspension. Maybe they have a high amount of RNases, and we see degradation happen really quickly, right? And so you end up being in this race against the clock, like Yuri mentioned, where you're trying to prep your samples as quickly as you can and then run them to the core or the lab that has the instrument you're planning to use. And the logistics behind that can be really difficult to try and overcome, right? If you're collecting samples out in the field, you might be even more limited. Maybe you don't have the option of cryopreservation in order to ship your samples back to the lab where you're going to be doing those experiments. And then additionally, if you're working with infectious samples, transportation can become even more complicated, right? And so all of these things sort of boxed together made us realize that we need to come up with an alternative to our traditional live cell assays that opens the door for customers to use different samples and different types of experiments than they've used previously. And so that's really what we're trying to do with the Flex kit. So by allowing customers to fix their samples, this allows you to do those time point studies. We can fix and store samples across different time points, wait until the experiment is completed, and then process those samples together to reduce batch effects in our study. Additionally,
One second,
yeah.
I'll give you a microphone.
Okay,
See if you can
perfect.
make it a little bit louder.
Yeah.
This up. This one. Should be able to a little bit louder.
Do it on this side. Can you guys hear me better?
Yeah.
Okay. All right, so like I was saying, the use of fixed samples as an input allows us to batch those time point experiments so that way we can save and store our samples and process them together in a single run on your Chromium instrument. So this is reducing batch effects. It's allowing you to multiplex, like Yuri mentioned, in order to reduce costs. And then obviously by using a fixation-based method, we're locking in our biological states at that sample collection point. For a lot of our samples, when we're using live cells, the stress of dissociation, of shipment, that's going to make gene expression changes that might alter the data that we see at the end, right? So we're looking at stress responses instead of everything we put into the experiment upstream of collection. So we're able to lock in those biological states as soon as we fix. And then additionally, we'll touch on this in more detail in a few slides, but there are certain cell types that don't handle dissociation very well. They're fragile, they're sensitive, they're going to lyse immediately upon dissociation. So by fixing these samples, we're actually able to retain those fragile cell types in the final data set. And then obviously from a transportation standpoint, once we fixed our samples, they're effectively dead. So we no longer have to worry about, transporting infectious samples. And then also we have more opportunity and sort of a wider range of temperatures that we can transport our fixed cells. We can use a fridge or freezer temp as opposed to requiring dry ice for our cryopreserved cells. So when we're thinking about our large scale multi-site projects, you now have sort of the ease and ability to have samples from different labs across the country, across the world, all be shipped to one lab where you can run your single cell experiments and reduce those batch effects in those large scale studies. So let's touch on the underlying biochemistry changes that we've made to this assay in order to make fixed sample input a possibility. And so the major change is that in place of doing a more traditional reverse transcription-based capture with our poly-A tails, we're now going to use a probe-based design. And so you can see here in this animation that the overall design scheme is that we are going to utilize what we're calling probe pairs. So there's a left-hand and a right-hand probe sequence. They're each going to have about 25 nucleotides of complementarity to their mRNA target. So the intention here is that we're increasing specificity and sensitivity, right? So specificity because only our properly hybridized left and right hand probe pairs will be annealed and then captured in the downstream library prep. So we're reducing background noise. And then as far as sensitivity goes, we have about threefold coverage across the body of your average expressed gene. For our higher expressed genes, we've reduced that a little bit so that we're able to capture the full complexity in your sample. Other things of note that just kind of make this easier for customers to use, if you are working with a clinical study because it's probe-based, we're indirectly capturing the presence or absence of our mRNA transcripts. We have this built-in de-identification. We're not capturing patient information like SNPs or CNVs that otherwise would make it difficult to transfer the data across different groups. And then like Yuri mentioned when he did the introduction, we have this built-in multiplexing. It's going to be based around this probe barcode you can see here on our right-hand probe. It's essentially a sample barcode. And so we'll apply that across all of the genes in our probe set to a given sample. And this allows us to multiplex our samples without having to do an additional library like you would with cell hashing. And then one additional note, right? We do have the ability to detect gene isoforms. So we've intentionally designed our mouse and human probe sets to capture these known isoforms. And then for folks who are interested in it as well, we also offer a custom probe design option as well. Okay, so like I mentioned before, using a fixed sample input really broadens the sample compatibility for this assay. So the different major types of samples that we'll touch on today and the best practices for preparing them will be fresh or frozen tissue, FFPE tissue, which having been at 10X for 2 years is still kind of mind-blowing to me that there's an option for you guys to work with those FFPE blocks and get single cell resolution. And then obviously with our more standard single cell assays, we'll also touch on single cell and single nuclei suspensions as input. Like I mentioned on the previous slide, right, we have this built-in sample-specific barcode. And this is really, I think, the power of the scalability of this assay. So currently what we offer to customers, you can scale up and multiplex 16 samples in a given Flex gene expression library, which would allow you to capture about 128 samples across an 8-lane chip if you're fully multiplexing that out. And then if we wanted to look sort of even further down at the number of total cells, we're going to capture over 1 million cells off of one of these chips. So to Yuri's point previously about cost, the more you put into the system, right, we can really reduce the cost on a per cell basis. And our sales rep is also here today, so if anyone's interested in talking about pricing and the cost, happy to have that conversation with you guys after the presentation. But really the take home here is scalability and flexibility, right? Like I mentioned, one of the aspects that I find to be the most interesting is the ability to batch and run samples within a study, especially if you're performing a clinical study. You might be getting samples coming in on a really irregular basis. It's cumbersome and time consuming to process them individually. So the best way to really go about it is to batch those samples and store them. And then once you have a full 16 or whatever the scale of your study is, you can take those fixed samples out of their long-term storage and process them in a single library prep. So saving you a lot of time as well as money. As far as the data, right, on this batching and running later, what we've done here internally is we've looked at different storage time points at different temperatures. So we have some options that allow you to store up to 7 days at 4 degrees. And then we also offer or have validated up to 6 months at -80. So what we did internally is we took a PBMC sample, we fixed and stored and then processed that sample at different time points in these different storage conditions. And what we're really trying to drive home with this particular graph is that across all those different storage time points, we're still able to reliably capture the different cell types within the heterogeneous mixture. So you aren't sacrificing the data quality by storing your samples for long term before processing after you fix them. Okay, so to zoom out a little bit on the overall workflow, we have our upfront fix and permeabilization steps, which is the bulk of what I'm going to talk about for the remainder of the presentation today, what those different protocols look like, and what are the best practices, sort of tips and tricks that we have for getting your samples ready. Next up would be that probe hybridization step. If we're using multiplexing, right, we'll apply different sample barcodes to each individual sample when we perform that probe hybridization. That allows us to pool and run those different samples into a single gem reaction on our Flex chips. Really simple and straightforward downstream library prep, which I'm sure the core is excited about for processing these libraries. And then like Yuri mentioned, the sequencing burden is going to be about half of what it would typically be with your 3' or 5' gene expression assays. And the reason behind that is the way we've designed the probes themselves. So we're reducing the presence of our really high expressors, so we're able to capture your low expressing genes without having to sequence as deeply. And then for folks who've analyzed data before, you'll still rely on Cell Ranger and Loop Browser to analyze your Flex data, just like you would any 3' or 5' gene expression data. So really briefly, I just wanted to touch on, we've talked a bit already how we can stop and store after fixing and permeabilizing our samples. Another unique stopping point that I like to highlight for customers is after pooling your samples from probe hybridization. The advantage I see here is, as anyone who's run a 10x assay before, you're going to load such a small portion of that single cell suspension that you have onto the chip. You now have the option of storing and keeping the leftover cells that you don't load on the chip, which to me is, I think, really useful. I'm sure there have been a lot of researchers in the crowd. Not every individual sample you process will necessarily be the one that provides a lot of biological insights, but every once in a while you kind of strike gold, right? You find this one sample in your data. Oh man, I wish I had more data points for this sample. This particular organism or patient really seemed to respond to this treatment. I'd love to see more data points. If you stored at this point, you can go back to that exact same sample prep. You've already hived your probes to those cells, and you can just run more of those cells in a new library. So it gives you a really unique opportunity to get more data for those really useful samples. Okay, so right now on market, we offer a human and a mouse probe set that are off the shelf available to be purchased. Additionally, if you're interested in developing your own probe set for your model organism, we do have contacts at IDT who are willing to work with groups, whether it be consortiums or individual customers who are looking to develop probes. We're happy to help support you guys with that. Yeah, Nelson.
How much cost and time frame is there to develop custom probes, like say mosquitoes that labs?
Yeah, it's a complicated answer. A lot of it has to do, right, with things like the genome build. Is there single cell data that's already available? A lot of that goes into how long it takes to physically design the probe set itself. And then as far as the cost, I believe IDT said that they would be willing to work with consortiums that spend, I think it's up to $100K starting, and then they would make it an off-the-shelf item that you could purchase from IDT. So the nice thing there is they're willing to work with whole groups. I think it's unlikely that an individual researcher would want to spend that much on a probe set, but if you have people in your field that are interested in going in on designing a probe set together, we can help you guys with the design part, and IDT is willing to work with you to make it something that you guys can order across the different researchers.
Okay.
Thank you.
Are you working on any new organisms to add to this at this point?
there's been some people who are interested. It's still early stages. I think for most academic researchers, right, it's having the funding and getting those numbers so that you can plan for your next grant proposal or whatever it may be. So for right now, it's mostly just in early stages, but I would imagine in the next year or two that we'll see more people who are ready to really pursue that option. Yeah.
Quick question. I just don't see the combination of fresh frozen tissue for those tomates. Is it there as well?
Yes, yeah, we're gonna go piece by piece. So actually, I'm glad you asked that question. So on the right-hand side of my slide, right, these are all the different sample types that are compatible with the Flex assay. Then we'll talk about best practices overall in the next couple of slides, and then we're gonna drill deeper into each one of these sample types individually, talk about what your different sample prep options are and why you might wanna choose one versus the other. So we will touch on that. Yeah. Okay. So let's just talk at a high level about sort of the logistics of things to consider when we're first getting started with Flex. So obviously the biggest thing is going to be sample input requirements. If we're starting with a single cell or single nuclei suspension, our general recommendation is to start with a minimum of 300,000 cells or 500,000 nuclei as input. If we're going from fresh or frozen tissue, we recommend starting with a minimum of 25 milligrams. And then sort of on the upper end, we validated our fixation reactions with up to 10 million cells or nuclei. So other things to consider about that sample input, if we're starting with a single cell suspension, we still want to have higher viability samples, so similar to our 3' or 5' assays that we've done in the past. So obviously the higher the viability, above 80%, is going to give us really beautiful data. But the really, I think, advantage of using Flex is that because it's probe-based, we can work with lower viability samples and still get data that's usable on the other end. And so internally, we've gone as low as 50% viability and gotten usable data out on the other side. And then the other thing that is, I think, really nice is that we've already optimized the fixation process. We offer a sample prep kit that you can purchase directly from 10x. And so this is finely tuned for our downstream systems. It's meant to make your life easier. You shouldn't have to do as much optimization as you would with your own homebrew fixation. And really the biggest consideration with the using fixed cells is going to be cell counting. And so to that end, most customers, I know I used to rely pretty heavily on Trypan as well when I was still at the bench. Most customers are using Trypan Blue to count their cells. Now, when we're using our Flex assay, our cells are going to be dead. And so the issue with that is that Trypan is going to be, obviously the Trypan Blue is going to stain all of our dead cells, but it's also going to stain the debris. And for anyone who's worked with Trypan a lot, it's a debris heavy stain to begin with. And that makes it really difficult to get an accurate dead cell count because we're going to be sort of inflating those numbers and including the debris that's staining with the Trypan in addition to our dead cells. So to that end, we're really recommending using a fluorescent cell counter in place of your typical Trypan counts. Propidium iodide is probably the most common one I've seen in the field. Ethidium homodimer also works really well. It's a little bit more expensive, so it just kind of, it's a decision on what makes the most sense for your lab. Both of them are going to stain DNA or RNA within the cell, and so it's going to be much more specific to your dead cells and not counting our debris. And with that, there are a couple fluorescent counters on market that I've seen quite a bit in the field. Happy to talk to people afterwards about sort of the caveats of working with each of them, and sort of if you're in the position to purchase a counter, we can talk about sort of what the best options are for that. Yeah.
So we have a Genomics counter. Will that suffice for the fluorescent thing? It has a little bit of a GFP, a blue light exciting, and it does the automatic picture imaging like this.
So I believe Ethidium homodimer will stain in that blue, it'll show up in that blue range.
Yeah.
I can look into it and just double check that the fluorophores would be compatible with the different dyes, but yeah, it's a great question.
What was the name of that one?
Genomics?
Yeah, it's gonna do some live looking for us.
Genomics Encounter or something. We have one in K2.
Yeah.
Would you be able to count it just on the setup?
Yeah, if you have a fluorescent microscope, that would also work. I have some customers who are doing that. I think the only drawback of the manual counting is if you're planning to scale up and really multiplex, there's going to be more counting steps with Flex, and so it's going to be more time consuming. The good news is your cells are already dead, so it's not like our cells are dying on ice while we're counting our other samples, but it's just going to be a little bit more work for you. Yeah. Any other questions on cell counting? I know this can be a bit of a bear for some customers, most customers. The good news is if you're running into issues, right, you can always reach out to me. We have a big group here at 10X. We're happy to help you guys optimize these steps. You don't have to struggle alone. Sample prep can be cumbersome, and we're here to help. Okay. So we've talked about how we're gonna count with our fluorescent stains, other things to consider when we're working with our fixed samples. Just like with any live cell assay, if you're noticing debris and clumping aggregation that you think could lead to clogs, at any point throughout the protocol, you can always just do a quick 30 micron filter. The 30 micron filter is really your friend for any 10X single cell assay. This is essentially the diameter of the microfluidics of our chip. So if your sample can make it through a 30 micron filter, what comes out of that is much less likely to cause a clog in microfluidics. Additionally, if you guys are looking to enrich for a certain cell population within a heterogeneous mixture, you can also perform cell sorting, and this can happen both pre-fixation or post-fixation. Again, I think the considerations here would be how hardy are your cells? Is flow sorting really stressful? Will the viability be an issue if you sort them while they're live? Those are the kinds of questions I would be asking myself if I was considering whether or not I wanted to sort before or after fixing my samples. We have quite a bit of guidance on how to sort our fixed samples. We just generally recommend using a nucleic acid stain like 7-AAD. And then on the other side of things, when we're talking about multiplexing, which if you're interested in applying for the grant program through the GSI and the single cell core, this is something you'll want to consider. When we're pooling samples together for multiplexing, we really want to be mindful of pooling similar samples together. So that either means same starting material. If we're using an FFPE block, we don't want to multiplex it with a single cell suspension, right? We know FFPE blocks are going to have a lot more degradation than our single cell suspensions. And so we'll see an uneven capture of those sample types in our final data set. Additionally, we want to be considering things like RNA content, right? Our primary cells, as opposed to our cell lines, are going to have a much different RNA content. If we're talking about a tumor cell versus your standard PBMC, the RNA levels are going to be much different, and so we want to consider those things when we're choosing what samples to multiplex together. The other thing to consider is how we're processing our samples for multiplexing. We'll get into the specifics of the fixation protocols in the next few slides, but essentially you can apply different fixation times. And so when we're doing a study across a multitude of samples, we want to apply that same fixation time to all of the samples that we plan to multiplex together. And additionally, even if you're single plexing, you want to treat them the same to try and reduce those batch effects. So I recommend picking a fixation time point that works best for you in your lab and then just applying it to all of your samples. Makes your life a little bit easier and you don't have to think about how each one individually was processed. When we get into multiplexing, the nice portion of the protocol is that we've allowed customers to pool samples together for their post-hybridization washes. And this is great because we, as people are probably already aware, having larger cell numbers makes it easier to pellet and wash cells without losing them. And so if we're multiplexing, you can actually go a little bit lower on the input for your probe hybridization, as low as 50,000 cells or 100,000 nuclei per hybridization reaction. And then the upper limit of those hybridization reactions is 2 million cells or nuclei. My general recommendation here is with any new sample type that you're trying out, I recommend going in on the higher end of these fixation and probe hybridization ranges, just so you can get a feel for what cell loss you're seeing in your own hands. Different samples are going to pellet better or worse than others, and then different people at the bench just have different levels of expertise with washing. So one person might see more cell loss than the next with their wash and spin steps. If you start with the maximum amount, you're more likely to have what you need on the other end. And then once you get comfortable with the assay, you can always scale down once you're really familiar with where you're going to see that cell loss. Okay. So we've talked sort of at a high level about fixation and the different sample types that you guys can use for your flex assays. So we'll go through one by one and talk about the different fixation protocols associated with each of those sample types. So the first one we'll hit on is just working with cell lines, primary cells, and nuclei. So anything in suspension that you want to fix would be applicable for this particular protocol. So I've included a link here that will take you to the support site where you can find this protocol, but some of the key takeaways are that we have tested this in-house with a slew of different suspensions, PBMCs, dissociated tissues. We've looked at DTCs, cell lines, nuclei, so we've really robustly tested out this protocol for you. Like I mentioned on the earlier slides, the lower end for this protocol is 300,000 cells or 500,000 nuclei. Other things that we've touched on a little bit, but keeping those fixation times consistent across samples in a study, using things like fluorescent dyes, and then in the back end of this user guide, you'll also find some guidance on how to set up your cells after they've been fixed for that long or short-term storage. Okay. Yeah, what's up, Nelson?
You still have to dissociate the tissue. You can't just fix a tissue directly.
So you can, but this protocol is for when you're working with a cell suspension that you want to fix. So I will touch on the tissue protocol in a couple slides. Yeah. Okay. So just an overall assay scheme, it's very straightforward. So you just take your cells or nuclei that you want to fix, spin them down, you'll resuspend them in fixation buffer. You'll apply that fixation reaction either for 1 hour at room temperature or 4C for 16 to 24 hours. So this is where you have that wiggle room of how you can fix your samples, and you want to apply the same fixation time and temperature across samples in a study. Once that fixation time is completed, we'll spin our cells down or our nuclei, and then we'll resuspend them in our quenching buffer. This is going to be really important because it's going to kill that fixation reaction so that we don't overfix and destroy our samples. And then from there, we will count, and you can either go directly into probe hybridization or you can store your samples until you're ready to move forward with your multiplex experiment. So again, just some tips and best practices. High viability, the higher the viability of your cells going into this fixation, the better the data quality. But you have a lot of more wiggle room than you do with 3 or 5 primes. You can go as low as 50% and still get something usable. We can always pass things through a 30 micron strainer if we're seeing a lot of debris or clumping during our fluorescent cell counting. And then we have specific directions in the back of this user guide for how you guys can store those samples for up to 6 months before you're ready to process them in a batch. Okay. We've talked about single cell and single nuclei suspension, so to Nelson's question, let's switch gears and let's talk about tissue. To me, I think that this is where most people who are considering flex are falling under, right? For anyone who's boxed into using tissue, it's cumbersome, especially if you're collecting it out at a collection site. Most people are cryopreserving or snap freezing that tissue and then bringing it into the lab. And traditionally with frozen tissue and our live cell assays, we've only really recommended getting nuclei out of that frozen tissue. Anyone who's worked with nuclei knows that nuclei preps are not easy. I think they're really difficult to work with. I wish that no one had to use them. And so this protocol that we're going to highlight here eliminates the need for a nuclei isolation from tissue. Okay, so high level key points about this sample prep protocol before we dig into the specifics. It's a universal protocol. So this is going to apply to all of the tissues. We have a really exhaustive list on our website that I can share with you for the mouse and human tissues that we've tested for both fresh and frozen with this protocol. Multiple stopping points throughout the fixation protocol outside of just storing your single cell suspension at the end for up to 6 months. And then generally we have a range of anywhere from 25 to 100 milligrams of tissue that we've tested for the different tissue types. And we can always talk more about what makes the most sense for your particular tissue type as you're getting into your study. Okay, so we've talked about how this is applicable for both fresh and frozen tissue. And so let's just hit sort of the overall workflow, and then we'll go through both fresh and frozen separately and talk about what your different options are and why you might choose one versus the other. So for the overall scheme, you'll have your fresh or frozen tissue, you'll chop it up into smaller pieces, and then you'll perform the fixation in those smaller pieces. This is going to be really nice because it allows us to lock in our single cells before going into dissociation. So this is where we can see retention of our fragile or more rare cell types. What's nice with this protocol is that you can stop in the middle. Essentially, once you fix those small tissue pieces, let's say you're at a clinical site or not in the lab collecting that sample, you can chop out that tissue, put it in the fixation buffer, and then you can ship it in that fixed small tissue state. Then you can perform that downstream dissociation back in the lab and process the samples when you're ready.
What do you use
For chopping, you want to use clean razor blades and glass dishes. And I'll highlight that in a couple slides. Reasons why you might want to go this route or use this protocol, we talked before, but some samples have really high RNase content, and so working with those in a live cell capacity and generating that single cell suspension can sometimes really impact data quality. So by fixing right away, we're activating those RNases and allowing you guys to capture more robust data in your final library. I already touched on this, but preserving the fragile cell types by fixing before doing the full dissociation. And then generally speaking, we see less clumping and better cell recovery using this method than some of the alternatives that we would have recommended for our live cell assays. The only considerations here or sort of caveats that might be an issue, right? You want to make sure that we're chopping into smaller pieces before applying the fixation buffer. We just want to make sure that we're fully fixing all the cells in the tissue. If it's not chopped into small pieces, we can't really guarantee that the fixation buffer is getting all the way in and fixing all of the cells. And then for some customers, the input requirements are also a reason why this might not be the most suitable option for you. Having more than 25 milligrams is sometimes not an option. So that's something to consider as well. Okay, so let's go piece by piece, and we'll start by talking about your different options for working with fresh tissues. This first option here in our flowchart is what I would consider to be the most common protocol that customers are using for live cell assays. They have fresh tissue, they immediately go into dissociating into single cells, and then you would use that single cell or nuclei protocol we mentioned in the previous set of slides to fix those cells and run flex downstream. The next option, if for whatever reason, maybe your cells are really large in diameter and don't physically fit through the microfluidics of our chip, so cell types that are bigger than 30 microns in diameter, you might want to do a nuclei isolation in order to make sure that you can reliably capture that information with our microfluidics. And so there are two different nuclei isolation methods. So we have essentially homebrew protocols that you could download off of our website and sort of bring in-house to optimize your own nuclei prep. We also offer a nuclei isolation kit that is purchasable through 10x Genomics. It's geared towards fresh and frozen tissues, and there's a list of tested tissues on our website, so if you're interested, I'm happy to share that. But for both of these options, we would take that nuclei suspension that comes out of those protocols and fix it using the protocol from the previous section of slides. And then lastly, specific to the chop fix protocol we're talking about here, you could take that fresh tissue, chop it into small pieces, fix those small pieces, and then do the downstream dissociation. So officially on our website, we call this the fixation and dissociation protocol. I like to call it chop fix. So if you hear me say that, that's what I'm referring to. So the key take homes here is there are a lot of ways to get to the end result of being able to use your samples with the Flex kit when you're starting with fresh tissue. So you just need to think about what makes most sense for the sample type that you're using, the amount of starting material that you have, and again, these are all things that I'm happy to help you guys work through, and would be happy to meet with you to discuss in more detail.
Sorry, Joanne, did you look at when it's frozen? Did you look at different storage times of this frozen tissue?
Yes, yeah, I don't know if I have the storage time slides for the tissue in this slide deck, but I can get you that information and send it to you. But yes, we validated that as well.
Is it reasonably?
Yeah, we're seeing the same for the stopping points. We're seeing the same coverage with the tissue as we were with our cells and nuclei protocol. Yeah. Okay, so let's also now switch gears a little and talk about frozen tissue. So like I mentioned before, frozen tissue is tough to work with our live cell assays because you really are boxed into using a nuclei prep. Again, you could use our nuclei isolation kit or your own homebrew method and then go into fixation downstream. My personal favorite would be to avoid nuclei if you can, because for some sample types, they're just really difficult to work with. And so you can take your frozen tissue, chop it, and then fix it, and this alleviates the need to do the nuclei isolation, and then you could go into your downstream cleanup steps for the flex assay. Okay, kind of looking at things across the different options that we just talked about, what are the advantages and disadvantages of each one? So for the nuclei isolation kit, depending on the tissue type, you actually might need more material in order to get that 500,000 nuclei out on the other end that you'll need for your single cell or single nuclei fixation. So it might make more sense to go through chop fix instead. And then the advantage would be that the kit that we offer is optimized for a lot of tissues. If you're working with one of those tissues that it's been optimized for, then this might make the most sense for you. For the chop fix protocol, sort of the disadvantages that we touched on is that 25 milligrams might be a lot of material for your sample type. That might not be an option for you. And then the other thing to consider, right, you have both the option of fresh and snap frozen, so a little bit more flexibility there. And then that dissociation in the middle of the protocol that we talked about, or excuse me, that fixation in the middle of the protocol before dissociation is what's going to allow us to capture those fragile cell types. And I have some data in the next couple slides to highlight this. And then generally what we've seen in-house is that we're getting better yields for cell numbers using this chop fix protocol. So it's worth trying out and seeing if it works for you. And then with the freshly dissociated samples, right, require looking at things like viability if we're making a single cell suspension. If you know your tissue might not be super viable to begin with, this might not make sense as an option for you, especially if you don't think you'll get 300,000 live cells out of that dissociation. But on the flip side, the advantages of dissociating fresh single cell suspension is that you may already have a protocol optimized in your lab from running 3 or 5' gene expression assays. So you already know how to dissociate and get a high viability sample, so it's just easier for you to do that, and that's perfectly fine. And then the other advantage is that because you're starting with a live cell suspension, you have the option to do things like antibody staining and sort of bead-based enrichments, back sorting, and other things that you might not have the option with starting with fixed cells. So those are sort of the high-level takeaways of pluses and minuses of each. And then, like I mentioned, we also have some data here to sort of take home the fragile cell type retention. So in this particular example, we're looking at mouse lung, and we're looking at a fresh dissociation. So making that single cell suspension and then going into fixation, using our chop fix protocol with fresh tissue and then using it with frozen tissue, all with mouse lung. And you can see here in the UMAP plots that while we do get good data with our fresh dissociation, right, we're seeing cell clustering, we're seeing different cell types within this lung tissue. If we compare it to the chop fix protocols and the right two plots, you can see this cluster here and this cluster here more or less aren't represented in the dissociation sample, or this little cluster here as well is pretty underrepresented. So I think these two plots really drive home that even with fresh tissue, the chop fix protocol does a really nice job of capturing those rarer cell types, especially in these heterogeneous tissues. So if you're working with a tissue type like this, it might make sense for you to use chop fix even if you could easily dissociate into single cells first. Additionally, kind of again looking at both the manual dissociation and octo dissociation, which I'm going to talk about in the next 2 slides. So essentially there's 2 ways you can run this tissue chop fix protocol. When we compare the chop fix protocol to the nuclei isolation kit, we see similar representation of more rare and fragile cell types in the final data set than we do in the nuclei isolation kit. This makes sense because with nuclei, right, we're relying on just the pre-processed mRNA that's left in our intact nuclei. So we anticipate seeing sort of less robustness with this type of a sample prep as opposed to using our chop fix. So just things to consider as you guys are making these decisions. And then really briefly, I just want to touch on, yeah, Nelson.
So a lot of cells seem to be gone from the nuclei.
Yeah.
Versus.
So it's mostly the immune cell population, which is pretty notorious. Most immune cells aren't really high expressing, especially if we're talking about PBMCs, your T and B cells. It tends to be a lot harder to capture robust because they tend to express really high at the protein level, but not have a lot of RNA. Things like CD4, TCR, BCR transcripts are really difficult to capture, but really highly expressed at the protein level. So it's common to see with nuclei.
Is there a way to address that? I mean, what's the reassurance to do the nuclei isolation if one starts losing so many cells?
Right. So I guess it's things like, do you care about immune cells? For some customers, there's just immune cells infiltrating their tissue, but they're actually more concerned with the tissue specific cells. And so if you're not interested in capturing the immune, then something like a nuclei prep would make sense. If you're working with brain tissue, cardiomyocytes, anything that are really, really large in diameter that won't fit through our microfluidics, a nuclei kit is still an option for you to be able to capture single cell without not being able to capture it all. It would clog the system. So, great question though. Any other questions on sort of the different options here? Okay. So briefly, I just want to walk through the steps of the dissociation protocol and the fixation protocol at a high level. So to Yuri's question, right, we recommend using glass to chop up your tissue prior to fixation. Seems weirdly specific, but using a plastic dish can actually generate plastic fibers that will clog our microfluidics. Anne's shaking her head because she's been involved in some of the troubleshooting cases. It took us months to figure this out. So use glass is the take home here. And then once you've chopped up your tissue, you'll use that fixation buffer to essentially rinse that glass dish and transfer into a tube for that fixation incubation step. Just like with our single cell or nuclei suspension, you can apply anywhere from 16 to 24 hours of that fixation time at 4C. And then downstream, essentially, we're just going to spin it down. We're going to quench that fixation reaction, and then we'll go into the dissociation portion of the protocol. Keep in mind, if oh yeah, here.
Sorry. So what's the definition of small pieces?
That's a great question. So being able to pass through a wide bore pipette tip is essentially what we're looking for here, so that you're able to reliably transfer into a tube. We're not leaving pieces behind.
Just like half a million.
Yeah. Great question though. So like I mentioned, if you're doing this portion of the protocol out in the field, you can stop and store those fixed tissue pieces here before continuing on with the protocol in the lab. And then the next step after going through that fixation, there are two different workflows for the dissociation. If you have a GentleMax octodissociator available to you, you can automate the dissociation steps by using a GentleMax C tube and adding that fixed tissue suspension into the GentleMax, running the protocol, and it essentially does the dissociation step for you. And then you take it off the instrument, perform some wash spin steps, and then you can count and move forward with the rest of the assay. For those of us in the crowd who don't have access to that piece of equipment, don't think you can't use this protocol, there's a manual dissociation step. Like you saw in the previous slide, manual and octodissociator reliably generate the same quality data with the Flex kit, so it's not the end of the world if you don't have the option to use the octodissociator. Again, it's just a little more work for you because you're doing it by hand. So if you're thinking about scaling up or really doing a large-scale study, you might want to consider purchasing or getting access to a dissociator. Yeah.
We do have open dissociator on the floor, so everybody welcome to use it.
Yep. So it sounds like that's available to you guys. The program that you would use is pre-programmed in there by the GentleMax system, so it's not like we have to put anything in that's specific to 10X. Okay. All right, so overall tips and best practices, just hitting the highlights of this section. If you're using a tissue that's not on the list of tested tissues, I always recommend performing a pilot study. Maybe you have a little bit less than 25 milligrams. It's worth seeing if you can get enough material out on the other end to still be able to use Flex, right? Things like tissue density can really impact whether or not you have enough material, so weight isn't always the end-all be-all in terms of the cutoff there. Things that we want to look for with our pilot study is just looking at cell yields, how much you're able to get out of a certain milligram amount of your tissue that you're using glass instead of plastic. We have this optional storage of the fixed tissue pieces if you want to do the rest of the collection in the lab. And then once we've gone through the entire protocol, just like with our single cell and nuclei suspension protocol, you can store those fixed cells for up to 6 months at -80, so you can still batch your samples together. Okay. The last sample input that I'm going to touch on is working with FFPE samples. And for most of the people in the crowd, you're probably familiar with formalin-fixed and paraffin-embedded tissues. It is sort of the end-all be-all, right? Most clinical sites only want to FFPE prep samples for you. And up until recently, this has been a real struggle for molecular work, right? Most people don't have anything that they can do with these FFPE scrolls. They've been sitting in a drawer collecting dust probably for years, hoping that, you know, technology would catch up and you could do something with them, right? And the reasons behind that and why they're so difficult to work with, the fixation is pretty harsh, right? We're cross-linking, it's difficult to get into our nucleic acids. We know that our nucleic acids are going to degrade, and it makes it really hard to make an NGS library. So that's typically what the hurdles have been. And then other things, there's not really standardization, right? There's a lot of different ways that you can embed your samples in these blocks. And so there could be quite a bit of variability of the actual quality of the tissue in the blocks as well. So the protocol that we've come up with has tried to address a lot of those issues and make it easier for you to still use those FFPE samples. So similar to our tissue protocol that we just went through, there's going to be a manual and an octodissociation version of how you can isolate single cells out of these scrolls, and we'll talk about that in a couple slides. There are quite a bit of guidance in the back end of the protocol that I can send to you that talk about the amount of input for the scroll that you'll need in order to get, you know, a certain number of cells out on the other end. And this takes into consideration the thickness of the scroll. And I like to point this out for people who are more familiar with histology. It's going to be a much thicker cross section than what you would typically put on a slide. So 25 to 50 microns in diameter is typically what we're recommending. And that's just so we're able to capture whole cells out of that cross section. The standard, you know, 5 to 10 micron cross sections that you'll see on a histology slide are just going to cut most of your cells in half. So like I said, in the appendix, we've looked at, you know, how much tissue is occupied by the block in terms of surface area, as well as the thickness and the number of the scrolls that you need to get a reliable amount of tissue out on the other end. And you can see down here, we've really tried to extensively work through different types of tissues. We've done a pretty in-depth look at different human FFPE samples, and we've looked at quite a few mouse as well. This protocol has been out for a little over a year now. I have a lot of customers that are working with it. People are really pushing the boundaries. I've seen some customers use blocks as old as 15 years old and get really beautiful data out of their isolations. So it's really impressive what people are able to do with this. Okay. So things to think about and consider when working with your FFPE scrolls. We realize that there's going to be a wide range of quality in these FFPE blocks, so we are anticipating seeing a slightly lower data quality than with your fresh or frozen tissue or your single cell or nuclei suspensions. We'll get into the specifics of the physical workflow steps on the next slide, but just like with anything else, we want to make sure that we're getting a really robust and fully dissociated single cell and single nuclei suspension. You can see it says both here. The sort of a byproduct of this protocol is it's going to end up being mostly cells, but some nuclei as well, just because of the way we're cross-sectioning through the scrolls. And the Flex assay is set up in such a way that we can take in that data, and you'll still see both of those in your final data set. Using a swing bucket rotor in order to increase the number of cells that we're retaining during our different wash steps, especially if you're limited to how much of the FFPE block you can use, that's going to be really important to reduce that cell loss during wash and spins. As far as handling the scrolls themselves, it's perfectly fine, especially most people don't have a microtome in their lab, so if you're going to a core that's doing that cross-sectioning for you, you can have them put those scrolls into an RNase or DNase-free tube, and then you can store that at 4C for about a week. So again, if you want to go to your core and have them, you know, make a bunch of scrolls for, let's say, 10 different blocks, you can have them do it all at once, bring it back to your lab, and process them as you're ready to. We generally want to avoid touching the scrolls once they're in the tube when we start the actual isolation process, so that way we are less likely to lose cells during that isolation protocol. Okay, so what does the protocol look like? So like I mentioned, we have an octodissociation method or a more traditional pestle method. The only reason we're going to call it out in this original deparaffинization steps is that if you're using the octodissociator, we're going to deparaffinize in the GentleMax C tube. We'll use xylene, a couple xylene washes followed by ethanol washes to deparaffinize and rehydrate the scrolls. And then from there, we'll go into the actual dissociation. If we're running this manually, you can use whatever tube is most comfortable for you, and then we'll use pestles to actually perform some of those downstream dissociation steps that we'll show in the next slide. So for the folks who are gonna go to the core and use the octodissociator, right, you'll use your GentleMax C tube. This is what you'll bring over to the core. You'll add in that dissociation enzyme mix, and then we'll run that on the GentleMax. Again, it's a pre-programmed protocol that's already in the system, so you can just push a button and walk away. Once that's completed, we'll transfer the cells from that dissociation into a new tube, and we'll immediately strain with a 30 micron strainer. We anticipate debris with our FFPE block, so that's why we intentionally built in that filtration step. And then we'll perform some washes and then downstream counting. If we're running this manually, this is where we'll bring out our pestle and we'll manually dissociate that those scrolls so that we can isolate out our single cells for downstream counting. Okay, so sort of the caveat to all of this, and I've touched on it a couple times, is I've seen 1-year-old blocks that are really terrible quality and then 15-year-old blocks that get really great data. So just the age of the block alone isn't always going to be a good indication of what is a good quality block for you to use. Generally speaking, we found that looking at things like a DV200 score can give you more of a sense of the quality of the tissue in your blocks. So for folks who aren't familiar, a DV200, you isolate total RNA, and you run it out on a fragment analyzer, and you just look at what percentage of that sample has RNA above 200 bases. And that is going to give you an indication of how degraded your sample is. Like we mentioned before, we know FFPE is going to come with a certain amount of degradation, and the way our probe set is designed, we expect to still be able to capture the presence and absence of those mRNA molecules. But if we have a really, really low DV200 score, that might be a sample that you would anticipate having really low data quality for. Okay. Like I mentioned, in the back end of the FFPE isolation protocol, our R&D team, bless their hearts for all the hard work they've done. They've listed all the tissues that they've tested, whether it was healthy or diseased, the size, so the thickness of the scroll and the number of scrolls that they used, roughly how much of that cross-section was occupied by tissue, and then what yields they were able to get for cells for both manual and octodissociation methods. So this is a really great jumping off point for customers when you're getting started. The good news is you can always run a pilot experiment. Those cells are fixed, so you can store them and then rerun that isolation again if you need to get more material, and you have up to 6 months at -80 before you get to that. So lots of wiggle room here. And generally speaking, the customers I've seen who run this protocol, it's very straightforward, easy to work with, and the data looks surprisingly good. And so to that end, I will kind of highlight some of the data here. And so you can see that by using this protocol on archive samples, we're still able to cluster out actually quite beautifully the different cell types that were in the FFPE block. For the folks who are heavy on the data analysis side in the crowd, one thing that you'll notice in our barcode rank plots, this is typically how we assess sample quality with our 5' and 3' gene expression. We expect that barcode rank plot to look different with FFPE samples. So if you're used to looking at your barcode rank plot and you're anticipating that steep kneeing cliff that you traditionally see with your 5', don't panic when you don't see it with FFPE. We expect it to be a little bit noisier. And so this is sort of the phenotype that we expect to see with these data sets. And then again, comparing the fresh frozen versus the FFPE isolation, really what we're showing here is that you're able to get comparable representation of the different cell populations using both approaches. I will say, generally speaking, if you have the option between fresh frozen and FFPE, I would go the fresh frozen route. I think that there's going to be more reproducibility and more consistency in the data that you'll get from that fresh frozen tissue. like we touched on with the FFP, the actual putting that tissue in the block, there can be a lot of things that can go wrong at that step that can be sort of an X factor, right? So here we're showing you a really clean block that has really good data, but that's not always the case in the real world. So if you have the option, I always recommend going the fresh frozen route. Okay. Just highlighting this again here, we've seen this a couple times at this point. Just wanted to remind everyone, right, we've talked about the different sample prep options that are available to you guys. We have multiple stopping points throughout the protocol. And then, you know, the core will be able to sequence those libraries at a lower sequencing depth for you guys. And then we can still analyze using the same software we've used in the past. So with that, I'm going to plug the support website as an FAS, right? We have tons of data, so if you want to see more of the data to some of the questions we had today about how Flex compares to other assays or how the different sample prep protocols compare to one another, there's a lot of data on our website that we can share. All of the protocols I mentioned today are up on that website. I have business cards that I can hand out. If anyone wants to email me directly and ask questions, you're more than welcome to. You can also always email support@10xgenomics.com. And with that, I will wrap up with any questions, and I will leave this QR code up. Again, I mentioned at the beginning, if you're interested in getting a copy of the slides from today's presentation, please scan the QR code. It'll bring you to a 10x landing page, prompt you for some information. It just lets me know that you'd like a copy of it, and then we can share that with you after today. Thank you.
You tested fresh frozen tissue in the OCT block and then cross-section that, kind of like a hybrid between.
Yeah. So there have been some customers who've started working with OCT embedded tissue. So we have a KV article on our website that I can share with you that kind of goes through the considerations. Basically, it's just running a pilot with the tissue dissociation protocol, making sure that it still holds well with your sample prep, and you would essentially start after fixation. So because you've already sort of fixed it with the OCT, you would go into the dissociation steps downstream.
Hold on a second, I have to run, but I just want to let everyone know the GSI's purchased food in the back, and if you could, grab food, continue asking your questions, that would be fantastic. And I appreciate everyone for attending, and hopefully this type of technology seminar has been useful to everyone.
Thank you.
Thanks, Nelson. Yeah.
So I have a question about the sample storage, because we have a number of freshly frozen human postmortem patient samples, but they have been in the freezer for a couple of years, several years. So I don't know if we can still use this sample for a single nucleus on sequencing. We have done PATA sequencing. I found there was some undegradation because only integrity number only around 5. So that means you are so.
Yeah. So I was gonna say the biggest thing that I've seen, it also depends how they're stored, right? Were they snap frozen and cryopreserved, or were they stored at -80 long term? A lot of those things can go into the tissue quality that you get at the end. So you can do prolonged storage, typically with cryopreservation, that tends to yield the best results. That being said, I mean, if these are samples that you can't get back again and you want to run them to get information out of them, I think Flex is still your best option to get information out of them. But maybe we could set up a time to chat and talk more about some of the ways you can do that. Yeah.
So I think we can still try. For example, like during the sample preparation process, for example, we can do a quality control to look at nucleus. If you have sufficient nucleus.
Yeah, we also, in the probe set, and I didn't touch on this today just because I didn't want to get too into the specifics, but we also have some mitochondrial genes that we've spiked into the probe set to look at sample quality as well. So it's sort of like an internal QC, because we expect there to be more mitochondrial RNA for samples that are really heavily degraded. So that's one way in the final data that you could look at that as well.
Before you address your next question, I did want to say that, so in addition to supporting the GSI with kits to run the pilot, so you guys will can apply for the pilot grant program. For anybody who's not eligible from a GSI membership standpoint, either not selecting because we can only select so many, or again possibly working with non-human mouse, I am offering discounts that to help kind of fund pilot projects as well. So reach out for that. Then ask Juliana the more questions.
If I understand correctly, it's supposed to be probe based. You will sequence the prop with the barcode rather than the real. Okay. So one prop designed for each gene, not each transcript.
It's about an average of 3 probes per gene. With our really high expressing genes, there's a little bit less probe coverage. And for our lower expressing genes, it's a little bit higher. So that way we get even representation in the final data. And that's where we get that reduced sequencing depth, because we can hit that complexity at a lower read depth. Yeah.
So how many genes will be covered?
So for human, it's 18,000 genes, and for mouse, it's 20,000. We also have all of the sequences publicly available on our website. So if you're interested, I can send you those CSV files, and you can look through them and make sure that the genes or the portions of the genes you're interested in are covered in there. And then, like I mentioned at the beginning, we also offer some guidance on designing your own custom probes, and you can spike them into the human or mouse probe set and capture, because we have some customers looking like viral genes or other things. I have one group that's working on trying to capture with CRISPR guide RNAs and fixed cells, so we can work with you to help you design something custom as well.
Last question.
Yeah.
About how well align this data with the previous, the 3 parameters.
Yeah. Yep. So we did do direct comparisons. We have some data that I'll send sort of a more exhaustive data package to Yuri, so that way he can share with everyone, or I can share it with you directly.
Okay.
But essentially, what we found is that this is a bit more sensitive than 3 or 5 prime. I think again, it goes back to the complexity of the library, not having to sequence as deeply to get the same amount of information, and so it's actually more sensitive than 3 prime. Yeah.
Thank you.
Yeah. No problem. Yeah.
So I noticed for your big sample, you would have the tissue fresh, chop it up and then fix it. So if you had, for instance, if you had tissue that was PFA fixed, like a whole brain that's PFA fixed, then cryopreserved in the fridge for however long, would our option still be to do the fixed protocol, or to just paraffin embedded and then?
So you could do either. I have some people who've taken PFA fixed and then they'll chop it up and run it through the chop fix protocol, just essentially skipping the fixation step. The risk you're taking there, and it's kind of up to you, is just you want to make sure that that tissue is really well fixed. So it's kind of, you have to use your best judgment on whether or not you want to run it through the 4% PFA fix that comes in that kit, or if you trust it's fixed enough where you can just chop it and go right into
So
the
we could do
fixation.
kind of like an extra fixation step with really.
Yeah, you could, right? The downside is like you can get into over fixing. And so it's one of those things where I guess if you have the tissue, I would try both ways and see what looks better. And then your third option would to be just to put it into like a block and then run the scrolls from the block instead.
Yeah, I guess it's still going to be the droplet, right? So the dipple site still won't be captured, right?
Say that again, sorry.
The dipple site still won't be with Flex, even it's fixed.
Yeah, I mean, outside of Flex, we also just launched some new chips for 3 and 5 prime. We're calling them Genomics chips. And for those, we've really changed the chemistry and the way the microfluidics works. And so now R&D is saying that you can go up to 200 microns in diameter with those chips. So that opens the doors for adipocytes, cardiomyocytes, neurons, things that we typically didn't recommend people run whole cell through, you theoretically could do with Genomics. So I can get you more info on Genomics as well.
How do you see using Flex to detect like the VJ rearrangement? This progress, you might have to design the probe.
Yes, I was gonna say that's the biggest drawback is for the immune people in the crowd. This isn't a great way to do VDJ discovery, right? Because we don't have a way to capture the entire intact transcript. But if you have a known sequence that you want to look for, you could absolutely, we already have some customers who are designing some CD3 like specific probes so that they can look for the presence or absence of like a known clonotype that they're interested in. But that's
on the discovery phase.
Yeah. On the flip side with Genomics, we now offer like a light fixation. So it's not going to be as robust of a fixation as it is with Flex. So you can really only store samples for about a week after fixing with that protocol, but then you would be able to use it with 5' Genomics. So that would be another option for you guys. If you're looking for like a way to sort of batch as you go, that I would push you that way. Yeah. Okay. I'm gonna stop this. Yes, absolutely.